Yes, so I am a postdoc with Nate Grubar at Yale. Thanks for having me here today. It's nice to talk about some arboviruses again. So I'm going to present some work that we've been doing on Eastern equine encephalitis. It's going to be a bit of a whistle-stop tour today. The original draft of this paper was like 10,000 words long. There's a lot of stuff in it. So if you're interested in any of the extra stuff, it's all in this paper. But okay, so a quick introduction to triple E because it's one of those weird little niche American encephalitises. So it lives mostly in birds. We don't know exactly which birds are relevant here, but probably small songbirds. It's transmitted between birds by mosquitoes, mostly Culiceta melanura, but there's a few different ones. And then it spills over into humans and horses, which are dead-end hosts because they don't produce a high enough viremia to pass on the infection. And it causes severe disease in both humans and horses with high fatality rates and lifelong neurological injury in the survivors. So mostly it's up in the northeast of the US, in this kind of regions. This is New York, Massachusetts, and Connecticut. Although it is present in, for example, Michigan as well. Michigan has quite a few cases, and kind of up across the East Coast, it's sporadic. But really, Massachusetts in particular, you can see here there's this kind of focus in the southeast of these three states, and then in upstate New York as well. So why do we care about it? In 2019, there was a large outbreak of triple E. When I say large, it's not large in the scale of a lot of outbreaks. In the Northeast, there were 19 human cases and 26 horse cases. Previously, and then across the whole country, I think there were about 38, and a lot of those other ones were in Michigan. So this was a big year, and obviously raised some concerns that these bigger outbreaks might be happening more commonly. So before we can ask if something changed in 2019, we have to understand what the normal dynamics are, right? So to answer this question, we turn to the genomes, because I am fundamentally a genomic epidemiologist. Triple E is a select agent, which makes it really hard to work with. And even the RNA is a select agent. So sequencing it is really tricky. Thankfully, the Wadsworth Center in New York State is able to work with it. So they sequenced 80 new isolates, 48 of which were from 2019. So you can see this big jump in the number of sequences there. That's pretty much all the Wadsworth. They're pretty much all from mosquito pools, these sequences, some from some exotic birds, some horses, things like that. And what's really good is that, so you can see from this map here, the stars are the newly sequenced isolates. Lots of those counties didn't have any sequences from them at all. So it really expanded our data set geographically as well as temporally, and just like the sheer number of sequences. So we use these to build a phylogeographic analysis. So this just means that we treated the sampling location of the viruses as a trait that evolves across the tree. And what we found in this time-resolved tree here that's colored by that inferred location is that Florida is the source of triple E in the US. So you can see that because the backbone of this tree here is all in Florida. So that means that it's spilling over from Florida to other parts of the US. And that's what this map shows here. These lines, the thickness is the frequency that you see them in the full Bayesian posterior. Just thick is more, right? So you can see most of it going from Florida up to New York and Massachusetts and Connecticut, and also Virginia, which is really interesting because we don't have a lot of data from Virginia. So there's definitely some unanswered questions there. Once it gets into the Northeast, these clusters are usually detected within like a year or two of introduction. That's what this plot shows here. And then they tend not to be super long-lived. So they're around for a couple of years in the Northeast. Some do definitely stick around a little bit more. There's a long tail on that, but fundamentally pretty quick. So we have 26 introductions into the Northeast in this time frame. And you can see three kind of rough waves here. So we've got like a group kind of here-ish, a group kind of here-ish, and a group kind of here-ish. It's a bit rough, but we think these waves probably refer to some underlying process in the bird immunity, which I'll kind of come back to a little bit later. And most of these are in one state, which you can tell by the colors. The mosquitoes don't move very far, and maybe once the birds are finished migrating, they don't move very much. I don't know, really not an ecologist, and I think a lot of people on this call are, so we can come back to that later if you're interested. There are 6 clusters which were sampled in multiple states. And that's these 6 pulled out here. You can see that it's not just that they're in like bordering counties, like there is some genuine movement. And 5 of the 6 start in Massachusetts and then go elsewhere. So even though multi-state spread is rare within the region, when it does happen, Massachusetts is a regional source. So next we want to ask what drove the outbreak in 2019, right? So we understand what the normal dynamics are. So first, the one lots of people were worried about, especially in this kind of post-COVID era where we have a lot of variants, is adaptation. So there should be some kind of phylogenetic signal of adaptation, even if we don't understand exactly what the phenotypic effects are. So this tree here is the time-resolved phylogeny of all of the EEE sequences that we have. So all of these ones at the end here are our 2019 sequences. You can see they're all across the tree, right? There are no unique shared mutations to those 2019 sequences. So this outbreak's not caused by a novel variant that's better adapted to infecting mammals. So that's a really good, like, first thing that we're able to rule out. We then tested a heap of other stuff and got no signal in anything. So you can see here, it's pretty much all in the same regions that have detected it before. Those stars are in areas which have already had them or like, you know, right next to it. Right next to it. There was no change in the case demographics in the humans. There was no change in the underlying dynamics of the virus. We tested whether humans were going outside more in 2019. They weren't. We did some stuff with bird banding data. We couldn't find any signal for that, although I will say we weren't exactly sure which species to look at. So there may be some signal there, but it's hard to pass out. So what else is left? The mosquitoes. So shockingly enough, it was also a big infection year within the mosquitoes. So the bars here are the infection rate. And you can see that in Massachusetts and Connecticut, where most of the cases were, it was a big year for mosquitoes. As compared to New York, which didn't have so many cases, it's a more normal year. And when we look at the number of positive pools against number of cases, they match pretty well. That's not super shocking, right? If there's more infected mosquitoes around, there's more cases. We started trying to look if there was anything different about 2019 specifically. So these 2 plots here show values from 2003 to 2019 with the red line in 2019 for mosquito abundance, infection rate, vector index, which is infection rate times abundance. And then we've got this index P, which is a modeled estimate of reproduction number based on climate factors like temperature, humidity, all of that kind of thing. So for index P, we can actually see that it's pretty normal for 2019, right? That red line's right in the middle of the pack. Like, it just shows that it's seasonal. But what's interesting in 2019 is that you can see that this infection rate kicks up early and kicks up quickly. So we actually have extremely high early vector indices in both states in 2019. And these were the highest vector index values in the entire 17-year data set. So when there's a lot of infected mosquitoes, there's a lot of human and horse cases. But can we predict when there's going to be a lot of infected mosquitoes? So we know that the climate factors, at least in the Northeast, don't have enough information in them because we can't predict that introduction of EEE into the Northeast from Florida. But what we tried to do is build a short-term risk model from the surveillance data in Connecticut and Massachusetts. So Toby Koch, who is a really brilliant, he used to be a research assistant with us. He's now a PhD student at Colorado State, built this model that took into account vector index, month of detection, the year, the month of the year, index P, all of these things to see if we could predict the cases. You can see here it actually, in Massachusetts, where there's more data, predicts it pretty well. And we've actually, like, even the big years too, it gets it pretty nicely. And the main thing that came out of this was that there was the month of first detection of EEE then predicts a worse season. But the turnaround time required here to make that useful is really fast, right? So you have to be doing detailed mosquito surveillance with a turnaround of like a week to get that positive result. So we would like to understand the longer term dynamics, but we actually can't yet do that. We don't know what the deterministic processes are in the long-term dynamics. And to understand that, we need some more information on the actual human prevalence. Is this a rare disease with a high infection fatality rate, or a more common disease with a low infection fatality rate? Because then obviously changes in the underlying transmission cycles are going to have different impacts on those two scenarios. The holy grail is really the bird data. We desperately need more information on which birds are relevant, what their immunity cycles look like, what their immunity to EEE looks like, these sorts of things. And finally, what's happening in Florida, because it almost certainly precedes large outbreak years in the Northeast, but we haven't had sequencing data for EEE from Florida since 2014. They're extremely busy as an arbovirus research unit. They've got much bigger fish to fry than EEE, but this is really one of our missing puzzle pieces. So just quickly to thank all of these people, the mosquito surveillance teams, because they do extremely solid work year in, year out. And this is Toby here who did the modeling work in this, and obviously Nate for supervising. So that's everything. Sorry, that was very speedy. If anyone has any questions, I'd be very happy to talk about it.